brain PET imaging agent is clearly a big need, particularly in the Parkinsonian field, and particularly for alpha-synuclein. The main advantage of having a PET tracer for alpha-synuclein will be basically to support the design and the running of clinical trial targeting this pathological protein. So this for sure can help patient enrollment to make sure that the right target is present, could help patient stratification, and of course, it could help having like a longitudinal follow-up to monitor the disease efficacy. And I think the availability of PET tracer, for example, for amyloid and tau, have clearly showed the path and what can be really like the use of those tracers to support clinical trial. Of course, there are certain synucleinopathies for which there is a clear need for a better and earlier diagnosis. And multiple system atrophy, or MSA, is clearly one of those diseases where usually the diagnosis comes very late and when the disease is extremely advanced. I think we have been facing the typical challenges which underline the development of a brain PET imaging agent. In addition, I think the main limitation is that alpha-synuclein is a target with a relatively low abundance. So this implied that the tracer has to be, that really have to show high affinity, they need to be extremely selective, and they really need to have an optimized signal-to-noise ratio. So the background has to be extremely low. Our current molecule, ICI-12-589, come from the screening of our small molecule library, which is called the Morphomer library. So this is a library of small molecular weight compound, which are designed to have CNS drug properties, particularly in terms of brain penetration. And they are designed to be conformation specific, meaning that they will preferentially bind to the aggregated or misfolded conformation of a certain protein, but they will not bind or minorly bind to the physiological or monomeric form of the protein. Particularly for the alpha-synuclein PET program, we really try to drive the medicinal chemistry optimization program using brain-derived alpha-synuclein from brain of patient, because we wanted to make sure that we were optimizing compound on the correct alpha-synuclein conformation or pathological conformation. So we really base our medicinal chemistry optimization strategy using mainly Parkinson's disease tissue section and tissue homogenate. And while we were advancing with the preclinical characterization, we were extending the in vitro analysis also to other synucleinopathies, as shown in the presentation at AAIC. This has included, for example, the dementia with Lewy body, the multiple system atrophy, and other form of Parkinson's disease, such genetic form or Parkinson's disease with dementia. And then we kind of follow the classical development of a PET tracer, looking at the proper pharmacokinetic profile, selectivity profile, off-target profile. After having taken together a very compelling preclinical package, we thought that it was really worth to evaluate ICI-12-589 into first-in-human study. And the first-in-human study was divided basically in two parts. The first part was run in collaboration with in vitro and was including 5 healthy volunteers and 5 idiopathic Parkinson's disease case. And then this study was extended through an investigator-initiated study with Professor Oscar Hansen at the Lund University and Skane Hospital, where he added to additional Parkinson's disease case, but mainly other synucleinopathy case, including few DLB case and mainly multiple system atrophy case. And the multiple system atrophy case is what has turned out to be the most interesting population for this tracer, because this is really the population where I think for the first time you could appreciate a signal separation from the other diagnostic group, including control and other synucleinopathies group. What was also interesting looking at the clinical images, so the scan of those MSA patients, is that we could pretty well differentiate MSA subtype C subject from MSA subtype P subject, where in the MSA subtype C, the signal retention for the tracer was mainly in the cerebellar area, particular cerebellar white matter and cerebellar peduncle, which is exactly the brain region where alpha-synuclein inclusion are expected to be in those cases based on what we know from post-mortem study. While in the MSA-P cases, so the cases that from a clinical point of view, they have more like a Parkinsonian phenotype and have mainly pathology expected to be in the basal ganglia area. This is where the tracer was showing retention. So it seems that with our data, we see a nice match from where the alpha-synuclein inclusion are expected to be based on neuropathological study versus what the in vivo signal looks like. Of course, we will continue characterizing with tracer in MSA. Of course, we are very interested in understanding how early alpha-synuclein pathology could be detected in patients with MSA. And of course, we would like to go back down to kind of the prodromal MSA cases. And the other key line of investigation for us, of course, is to understand whether this signal is evolving over time in order to have like a longitudinal follow-up of those patients. Of course, we will also further characterize other synucleinopathy case. And in terms of discovery, we keep working on new molecule that, by having improved binding properties to alpha-synuclein, might allow also to detect alpha-synuclein pathology, potentially in idiopathic Parkinson's disease.